part 2 of online lecture 3. In this lecture, we'll discuss the structure and function of the most abundant class of macromolecules, the proteins. I think proteins are absolutely fascinating. They are by far the most diverse of the biological macromolecules. Their functions are many and really too numerous to mention in this short lecture that I'm going to give today. I'm just going to highlight a few of their most common functions. We discussed enzymes in the last chapter without really talking much about the enzymes themselves. Well, it turns out that most enzymes are actually made of proteins. And of course, we know that the role of an enzyme is to act as a biological catalyst which speeds up a reaction. This slide is showing an example of a protein enzyme called adenovirus protease. This enzyme aids in viral infection, and during that process it actually allows for the replication of viral DNA. It's so important that if this enzyme is actually inhibited, you can prevent the infection by this virus. Proteins often have a structural function. For example, as in collagen. Collagen actually has a number of functions. It's found in connective tissue, for example. But it's most well known for the role it plays in the support of the skin. Of course, it's well known because this collagen actually degrades during the human aging process, and it's the reason why some folks have been known to have collagen injections to try to bring back some youthful suppleness to the skin. The proteins myosin and actin are involved in movement, for example, as in muscle contractions. Here you can see the protein myosin attaching at different locations along the protein actin. Together these act to bring the actin closer to the sarcomere and actually cause a muscle contraction. Proteins are also involved in cell signaling. You may have heard of this one, EPO, that stands for erythropoietin. It's a naturally occurring protein, but a lot of people have heard of it because it was used as a performance-enhancing drug by athletes quite a bit in the past. And they used it because it increased their red blood cell count. And this is important to increase their oxygen carrying capacity and can result in improved athletic performance. In the body, EPO acts as a signaling molecule or a hormone. It binds to another protein called EPOR, or the EPO receptor. And here we see another example of a protein function. So in this case, EPO is actually a signaling molecule that binds to a protein, EPOR, and this receptor is acting as what we call a signal transducer. Signal transducer gets their names because they take a signal that is external to the cell and they transmit it internally. So what happens in case of these two proteins, the EPO and the EPOR, is that when the EPO binds this receptor, it can lead to an increase in or what we call a proliferation of the red blood cells. And we've talked about then how proteins can play a role as catalysts, as structural compounds, as hormones. They play a role in movement. They also can serve as receptors. But these are only a few. They actually have many more functions than. In terms of their chemical structures, proteins are polymers of monomers called amino acids. All amino acids have the same basic shape, which consists of a central carbon atom joined to a hydrogen, an amino group, a carboxyl group, and an R group. The R group varies from one amino acid to the next and determines the chemical characteristic of the amino acid. Thus, the general structure of all amino acids is the same, but only these R groups or side chains vary. There are 20 amino acids placed into 4 groups on the basis of their chemical structures. The first of these are the amino acids with charged side chains. 3 of these are positively charged, and 2 of these are negatively charged. all readily form hydrogen bonds with water and thus are considered hydrophilic. In this slide, the white boxes indicate the side chains. Notice that the rest of the structure is invariant from one amino acid to the next. Next are the amino acids that are not charged, but are hydrophilic due to the presence of polar side chains. There are 5 of these amino acids, and I'd like for you to stop just a moment and see if you can look at those amino acid side chains and determine what causes the polarity of those particular groups. If you remembered from the last chapter that differences in electronegativity between two atoms that are greater than 0.5 result in polarity, you're on the right track for answering that question. And if you remember that, you might remember that bonds that involve oxygen and hydrogen, or nitrogen and hydrogen, can lead to polarity. There are 3 amino acids, cysteine, glycine, and proline, which do not fit neatly into any group. So they're what are referred to as the special cases. Cysteine is special because of the presence of what's called a sulfhydryl group. And these are very important because these sulfhydryl groups can form a special kind of covalent bond with other sulfhydryl groups to form a structure that's called a disulfide bridge. And these are very important in forming the tertiary structure of proteins, which we'll look at in just a moment. Glycine is special because it only has a hydrogen atom as its R group, which makes this a very small amino acid that fits well into tight spaces. Finally, proline has a very unique ringed structure, which doesn't allow it to form bonds that result in a special kind of secondary structure called an alpha helix. We'll look at that secondary structure in a moment as well. The reason for this is that proline can't form hydrogen bonds, and so it's referred to as the helix breaking residue. And in terms of the overall three-dimensional shape of a protein, these prolines tend to be found in the bends and the loops of those molecules. Finally, there are 7 amino acids that have nonpolar side chains, and these tend to cluster on the inside of proteins away from the watery or aqueous environment of a cell. Proteins are formed when condensation reactions join amino acid monomers through a covalent bond called a peptide bond. These form the protein backbone and result from covalent linkages between the carbon of one amino acid and the nitrogen of another. Proteins always form or are synthesized in this direction, so we refer to the direction of proteins as one that proceeds from the N direction to the C direction. And if you look here on the bottom of your slide, you'll see that the N direction is what's referred to as the N terminus, and the other direction is referred to as the C terminus. And this orientation, this N to C direction, is analogous to that 5' to 3' synthesis that we saw in the nucleic acids. Now we're going to start to take a look at the overall structure of proteins. There are 4 levels of protein structure: primary, secondary, tertiary, and quaternary. And we're going to discuss each one of these in turn. The primary structure of a protein refers to the precise sequence of amino acids that are held together by peptide linkages. The primary sequence is very important because it governs higher levels of structure, because the properties of the R groups, those chemical properties that we just looked at in the previous slides, they actually determine how proteins twist and fold. So let's take a look at some examples of primary structure in a protein. As I said before, this refers to the precise sequence of amino acids. And this can be written in a couple of different ways. Sometimes you'll see it written as is shown here in a 3-letter code, where each of these 3 letters stands for a different amino acid. In this case, this is a 4 amino acid chain, which consists of methionine, valine, arginine, and cysteine. There are also one-letter codes for each of the amino acids, and I won't pretend to have them memorized because I don't. But if you ever see a sequence that's written that looks something like this, looks quite different from the A's, T's, C's, and G's that we saw in the nucleic acid DNA, you'll know you're looking at a protein structure. This is actually a protein sequence for a protein called bovine ribonuclease, and it's just a very short portion of that entire protein. This slide is showing another depiction of the primary structure of an amino acid. And what you can see here is that the precise structure of the R groups is actually not indicated. The R groups are shown with the brown balls here, and they're just indicated by R's. And you can get a very good sense of what the protein backbone looks like, and every red bond that you see indicates a peptide bond that formed between two amino acid monomers. Because there are 20 amino acids, and proteins can get quite long, there's an incredibly large number of potential protein structures. For example, just in terms of a dipeptide, this is a short protein, it's 2 amino acids long. Because either of those 2 amino acids in the dipeptide can be any of one of 20, to determine the possible number of dipeptides that you could have, you would multiply 20 times 20. So there are 400 possible dipeptides. If you scale that up to a tripeptide, you would calculate that by 20 times 20 times 20, which gives you 8,000 possible tripeptides. And the number gets even larger. By the time you get up to a protein that's 100 amino acids long, the number of possibilities in terms of structures outnumbers the number of electrons that there are in the universe. Yes, I just said electrons. I'm going to say that one more time. So for a protein that is 100 amino acids long, there are more possible structures than there are electrons in the universe. The point being that because there are 20 amino acids, and because proteins can get to be very long, and because the precise sequence of these amino acids determines the actual structure of a protein, the number of possible protein structures are virtually limitless. It's extremely diverse. So in order to start to understand how this primary structure of a protein gives rise to other structures, we need to look at the next level of organization, which is called the secondary structure of a protein. The secondary structure of a protein refers to regular, repeated spatial patterns that occur in different regions of the polypeptide chain. There are 2 primary types of secondary structure. The alpha helix, which has a structure that looks very similar to the coiled structure we saw in DNA, and the beta pleated sheet, which is unlike anything that we've seen before. Both the alpha helix and the beta pleated sheet result from hydrogen bonding between near neighbors. These hydrogen bonds form between the carboxyl group of one amino acid and the amino group of another. And this forms what we call a localized structure. So it happens between atoms that are fairly close together along the protein. The next level of protein structure that we're going to consider is the tertiary structure. This refers to the 3-dimensional structure that's formed by the bending and folding to give the overall 3-dimensional structure of the protein. This is unique to a particular protein and is also a function of the sequence of amino acids. In addition to the sequence of amino acids, there are other kinds of bonds that help form this 3-dimensional structure. Remember when we talked about cysteine and the fact that it formed a special kind of bond with other cysteines? I referred to these disulfide bridges. In this diagram, this yellow bar shows an example of one of these disulfide bridges, and you can see that it actually helps stabilize a structure between an alpha helix part of a protein and a linear part of a protein. In addition to the disulfide bridges, hydrogen bonds, hydrophobic interactions, ionic bonds, and a weak force called a van der Waals force are all involved in this three-dimensional folding of a protein that gives rise to its tertiary structure. And unlike what we saw in the secondary structure, this tertiary structure actually results from interactions between the R groups. So, for example, between the two sulfhydryl groups to give rise to those disulfide bridges. So just to recap here, the secondary structure is actually formed by interactions between a carboxyl group of one amino acid and an amino group of another, whereas the tertiary structure results from interactions between R groups. The last level of protein structure that we're going to consider is what's called the quaternary structure. The quaternary structure refers to the interaction of different subunits. A subunit is actually a unique polypeptide, and in the quaternary structure they actually come together and form the larger structure of the protein. Now each of these, if you look at this diagram, you can see subunit 1, 2, 3, and 4. Each of these is a separate polypeptide with its own three-dimensional structure. And these are held together by some of the same kinds of bonds we saw involved in shaping the tertiary structure. Here's an example of a quaternary structure of a protein. This slide is showing the protein hemoglobin, which is very important for carrying oxygen in mammalian blood. It is formed by two alpha subunits, which are shown in blue, and two beta subunits, which are shown in green. Hemoglobin also contains an iron-rich group called. These subunits are held together by a variety of different forces: hydrophobic bonds, hydrogen bonds, ionic bonds. These bonds are relatively weak, and small changes in the shape actually help in the function of hemoglobin and allow it to carry oxygen. In fact, when oxygen binds to hemoglobin, ionic bonds are actually broken. This is in stark contrast to the covalent bonds, which form the polypeptide chain and are very strong. So the primary structure is held together by much stronger bonds than the secondary, tertiary, or quaternary structures. The shape and chemistry of proteins are very important in determining their function. For example, the type of R groups that are exposed actually govern what kind of chemical interactions can take place. For example, if hydrophobic side chains are exposed, then you wouldn't expect that particular protein to participate in a lot of hydrophilic interactions. This slide is showing a very important example of protein structure and how it affects protein function. We're looking here at a protein that's called the sliding clamp. It looks like the gray donut that's shown here. And in the middle of the gray donut, this is actually an alpha helix of DNA. This sliding clamp is very important during DNA replication, and it's part of what keeps the enzyme responsible for building new copies of DNA from falling off the DNA while it's doing its copying. So you can see that this looks very much like a lock that locks around the DNA. The drawing in the upper left-hand side of your screen here is actually of the same structure that you're looking at on the right. This is the sliding clamp. This time it's actually drawn with what we call a ribbon diagram, and you're looking down the center of the DNA molecule. Whereas over here, the sliding clamp is shown by a space-filling diagram. In addition to the shape of the sliding clamp, it also has positively charged amino acid side chains on the inside. And these are very important for helping it interact with DNA, which has an overall negative charge. The proper folding of a protein is vital for its function. In fact, errors in folding can actually lead to human disease. This slide shows an example of a protein called amyloid B. The left-hand side of your screen here is showing the normal folding of a protein. On the right-hand side of your screen, you can see the misfolded protein. This misfolded protein causes lots of problems for people who have it because they aggregate together and form what are called plaques. These plaques are what have been found in the brains of Alzheimer's patients. So obviously there's a lot of work going on to figure out what causes these proteins to misfold and whether there can be a treatment for it. Sometimes a protein that is folded can actually lose its shape. And this is what's referred to as denaturation. So this diagram is showing a properly folded native protein on the left-hand side of the screen and the unfolded or denatured protein on the right-hand side of the screen. And what you'll notice about this is that the amino acid chain is still intact. Remember, I told you it has very strong covalent bonds holding it together, whereas the secondary structure, the alpha helices and the beta pleated sheets, and of course the overall three-dimensional structure, the tertiary structure, those have all been lost due to denaturation. Sometimes this process is reversible, and sometimes it's not. Denaturation can be caused by high temperatures. It can also be caused by exposure to certain chemicals. And in fact, your body uses the fact that enzymes can be denatured as a defense against bacteria and other kinds of infectious agents. So if you've ever run a fever when you've gotten sick, the reason for that is by increasing your internal body temperature, there's a possibility that you'll actually denature the enzymes of some bacterial pathogens. So a lot of people will say to try to let a fever run its course to the extent that you can, because that's actually your immune system working and doing what it's supposed to do. Oftentimes the denaturation of an enzyme or protein is not reversible. For example, like frying an egg. I'm sure you all know that once you've fried an egg, you can't unfry it. The reason for this, in the case of frying an egg, is that amino acids that were previously buried have become exposed and a completely new structure has formed. And there's basically no going back. So this completes part 2 of lecture 3. In part 3, we'll come back and have a more detailed discussion of some of my favorite proteins, the enzymes.